से इसमें हूँ, जूम में हूँ सर मैं। आई एम देयर इन द जूम एंड दैट इज नॉट वर्किंग। सर
नमस्ते मेयर सर। Namaste sir, are you able to hear me?
Yes, Dada, you're loud and clear.
Yes sir.
This is Dr. Mir.
Yes sir.
हेलो सर, I'm audible?
Yes, Mir Dada, looking smart.
सर जी नमस्ते। I'm audible
Namaste.
sir?
Yes, very clear like always.
ओके ओके सर, YouTube का थोड़ा प्रॉब्लम है।
नहीं हो गया ना?
हाँ, वो और एक लिंक है, उसको भेज रहे हैं सर।
It is showing live you on YouTube.
कल से सर ये प्रॉब्लम नहीं होगा ना? चलो सर, विल बी स्टार्ट बाय 3:30।
3:35, 5 minutes buffer you give. First,
Okay.
first session, no? Since president has to talk and inaugurate and you have to introduce. So
Yeah.
my request 5 minutes. Huh?
Okay.
Thank you.
Thank you. Hello. Hello.
Hello.
Can we get
Am I audible, sir?
started, sir?
Yeah.
Yes, sir,
Yes,
you are.
yes.
So good morning, ladies and gentlemen. Welcome to Odisha. Dear participants, it's raising up. So once again, on behalf of Odisha Pharmacy Teachers, Association of Pharmaceutical Teachers of India, APTI, Odisha State Branch, I would like to welcome you all dignitaries uh, to this innovative program, Faculty Advancement Initiatives by APTI Odisha, in support with the APTI Central Team. I would like to thank all of your participation teachers, because in this period when the whole world is suffering and the pharmacy teachers are also getting suffering in the COVID-19 pandemic lockdown, but still you have find out time for your learning processes, and Apti Odisha is happiest gratitude offering to you all. At the same time, you'll be finding lot of people on the dais who used to be sit and speak on the dais, give the deliberations. They all are here joined with us to support Odisha pharmacy teaching community and to be with this program. So on behalf of Apti Odisha office bearers, whole pharmacy fraternity of Odisha, I pay my sincere gratitude to all of you. So today's topic is PK modeling of targeted and novel drugs, as you all know. And before to that, my whole office bureau had a discussion and talked that when everything is not in a good situation, so our inauguration should be also very brief, and we should focus more on the beautiful lecture, beautiful presentations. So before spoiling more of your time, I just want to go to straight way to our honorable president, national president, Dr. Chaudhary sir, Praveen Digaman Chaudhary, and will inaugurate this session. So a big clap for the national president. And sir, welcome with all of your support. Association of Pharmaceutical Teachers of India is doing fantastically well during last 4 years. And Association of Pharmaceutical Teacher of India, APTI, is not only making a platform for all the teachers for better learning and teaching advancement, but also addressing to the different issues of the teachers, different issues of the institutions, different issues of the academic system, and trying its level best to call upon to everybody, making sit each other and to solve a problem and to find out a new way to the system. So, in this condition, APTI Odisha is going for some innovation programs, and you being the facilitator, modulator, I welcome you, sir. I just like to have few words from you so that we can directly go to the session. Thank you, sir.
Thank you. Hello.
Yes, sir. You are audible.
Am I audible?
Yeah, audible,
Mir,
sir.
how much time shall I? I
Sir,
have.
few minutes will be better, sir. By maximum to 3 minutes, we will start, sir.
Okay. Okay.
Thank you, sir.
Okay. So, ladies and gentlemen, very good afternoon to one and all. It really gives me pleasure to welcome you all on this occasion of e-faculty advancement initiative organized by PTI Odisha State Branch. At the outset, I would like to congratulate Dr. Mihir Kar and the Vice President Secretary of APTI Odisha team for taking this initiative and organizing this wonderful event of EFAI on road ahead scope of online teaching and advancement. Dear colleagues, we are going through difficult and tough time, and it is very essential to take care of ourselves and our beloved one. On behalf of APTI core team, I wish you a very happy, healthy life ahead. APTI has recently celebrated its Golden Jubilee in 2016 and for completing this 50 years of its existence. At this juncture, it is really imperative and indeed obligatory to state that we are greatly obliged and highly indebted late Professor M.L. Sharaf and late Professor G.P. Srivastava and other who are pioneers to establish this association way back in 1966. They have envisioned the promotion and pursuit of excellence in pharmacy education, research, promotion, profession, and to serve this cause of community at large. And the consistent and concerted efforts of their strong commitment, determination, dedication, devotion of all these stalwarts, which enabled APTI to strive to scale a new heights since beginning. And in this course of time, APTI has taken a significant stride on several fronts to make the holistic development. Currently, APTI is having almost 23 state branches and 2 international branches. One is at Malaysia and Turkey. And we also provide ideal forum to bring together all pharmacy leaders, teachers, researchers, industry people, government and private sector, university people, pharmaceutical entrepreneurs, medicinal and healthcare providers, and scholars of interdisciplinary and multidisciplinary sectors to discuss, deliberate, interact, and share their views on the topic of current interest in order to explore the new vistas of different disciplines of pharmacy and allied healthcare. Friends, APTI, the main motto of our association is to update and upgrade our life members as a teacher with the current and contemporary issue, and that is the motto. During this lockdown period also, we have organized several FDPs and several webinars, which are definitely thousands of teachers from all over the country got benefited through all these webinars and FDPs. Few more are in pipeline, and you will get soon, very soon, you will get the notification for that. I thank from bottom of my heart to all the pharmacy teachers for giving an overwhelming response to these webinars and FDPs organized by APTI. APTI also ponders to conduct various state, national, and international conferences every year, and we have conducted recently 5th MESMAP at Turkey in the April 2019, and 6th conference, international conference on pharmaceutical R&D and biopharmaceutics, with the help of collaboration with the BioLigas at Kuala Lumpur, Malaysia. And we are also lay stress on security safety of the pharmacy teachers, and the association has provided accidental death insurance cover for all every life members to the tune of ₹3 lakhs. It is also a matter of great pride that APTI gives top priority to women empowerment. The association has enabled the separate women forum for the academic growth and development of women pharmacy teachers. APTI also releases women forum bulletin in order to portray the outstanding contribution of pharmacy women professional and pharmacy teacher. The association has also instituted a special award for APTI Women of the Teacher, and which will be delivered in the national convention every year. APTI also promote research culture for the young and aspiring teachers and academicians, and to carry out this high and patentable research. In this regard, the APTI has published Indian Journal of Pharmaceutical Education and Research, Indian Journal of Pharmacy Practice, APTI Bulletin, and Women Forum APTI Bulletin every after 3 months. And these are our official publication. IJPER, friends, it is well distributed in 40 countries, and it is a Scopus and Web of Science Index journal having an impact factor 0.5. Besides this, APTI also conduct various conferences. We are also publishing more than 20 books of our own author, namely Padma Shri led Professor Harkishanji, then S.K. Kulkarni, Dr. S.K. Kulkarni led Professor B.D. Miklani, and many more. Friends, education is a vital to pace of social, political, economical development of any nation. So effective teaching is very essential. Effective teaching is important because teaching is based on helping children progress from one level to the another in a more sociable, interactive environment to get approach right to get student to be independent learner. And effectiveness does not mean that being perfect or giving a wonderful performance, but bringing out best in the students. And that is the main objective of this e-faculty advancement initiative, and this is organized by the APTI Odisha band. The main objective is to deliver the learning context effectively and interactively to the student, to increase the interest of student in learning the subject online, and to offer a flexible learning to the student, to make learning more student-centric and accessible. Last but not the least, hope this EFAI on road ahead, a scope of online teaching and advancement would bring a new zeal and ecstasy in the faculty participant and who are participating from all over the country. And I'm confident that all meaningful discussion, fruitful deliberation, broad interaction, and sharing of views in this EFAI would certainly give an impetus to excel further in your domain. Thank you for patiently listening. I profusely thank Dr. Mihir Kar, the President, APTI Odisha State Branch, Dr. Somejit Panda, Dr. Snigdha Patnaik, the Vice President of APTI Odisha, for giving me this opportunity to interact with all the participants. On behalf of APTI Central Team, I profusely thank all the resource persons of this e-Faculty Advancement Initiative program who are contributing by updating our pharmacy teachers. I wish a great success to this EFAI. Thank you so much. Over to you, Mihir.
Thank you, sir. Like always, refreshing, speaking from the heart, the young but the dynamic president of PTI. We are proud of your efforts, sir, because the whole Indian teachers have witnessed the strength of APTI during this lockdown period. We are reaching every night with thousands of teachers, whole India, and fantastic interactions and all this going on. And APTI Odisha also get inspired with all this effort of yours and started this 4-day learning program for its targeting. Basically, we have targeted initially to our teachers of Odisha, but right now more than 70% has come up from all over India. So on behalf of Odisha, APTI Odisha, once again I thank you, sir, for this opportunity. I thank all of the central committee members who are present. It will be not easy to take their name right now, but thank you all central committee members. Also state presidents are also there. It's very nice that whole Indian presidents and vice presidents are standing with APTI Odisha. It's a great gesture of yours, sir. We are obliged, along with none of other than any state or any pharmacy education cannot lead with the support of the stakeholders and some seniors, professors, academicians, deans, directors. So thanks a lot, Dr. Manoj Das, Odisha Pharmacy College Association Secretary, and one and only Dr. Shruti Ranjan Mishra. Thank you, sir, for being with us and supporting the events continuously since 2012. It's a nice gesture of yours, sir. We are obliged. Along with Vandana Madam, thanks a lot, ma'am. You have found out some time for us. We really appreciate your effort and we are really obliged to your learning advancement program suggestions and guides to you, to us, all of us. So without spoiling more of time, it is the time, sir. This topic we had once in the webinar series also, but in a different way. It was tossed so high that all of my pharma teachers asked that we want this. And Dr. Dan told that, okay, the secretary sir told that, okay, I can give you the recording. Manola, no, sir, once again we want live. So once again, welcome Chandramouli sir. So over to Chandramouli sir.
Namaste, sir. Namaste. Thank you for having me.
Yes, sir. Over to you, sir.
It's really an honor to be in the EFAI program organized by Odisha chapter. One of the important things which gives me goosebumps is that it's not a week ago we had a huge cyclone, Amphan, which is ravaged Odisha, but the resilience of Odisha people is really exhilarating. Thank you for all the hard work in spite of all your difficulties you have put to organize this effort. So I'll be just starting with the first topic of discussion today, which would be a very contemporary topic in the sense because many of us as pharmacy teachers are doing lots of research in academic setting. So what we need to do is that many of our research activities are based on devising drug delivery systems for improved performance characteristics or even better biological responses. So this talk is basically modeled to give you certain important recent breakthroughs that has happened in the domain of targeted and novel drug delivery systems. What I meant by targeted or novel delivery system is that for this purpose of talk is that any drug delivery system which is going to be organ specific, which aims to deliver the drug to a particular organ, or the release characteristics of that delivery systems are going to be modified in some ways so that it is going to be achieving some biological responses, are defined as targeted or novel drug delivery system for this purpose of this talk. So the outline of today's talk would be, we'll be having a basic introduction on how this domain has progressed in the last 5 years. Second, we'll be talking of the PK models, how the domain of pharmacokinetics has evolved in the last 43 years. And then we'll be talking of the recent advancements in the PK, especially the ACAT model and the extensions. And then we'll be having a brief overview on Gastroplus, a very important tool which is available in the domain. And then we'll be going through 3 case studies specifically targeting pulmonary, transdermal, and oral cavity. And then we'll be taking the talk. So before we delve heads on into the talk, while researching for this talk, I found that there is a very distinct paradigm which is emerging in the last 5 years or so. So whenever you see majority of the publications which has happened in this targeted or novel drug delivery systems, many people are resorting increasingly to the use of quality by design, because this is going to be a very important approach or framework for agile and more flexible drug development. So this is one of the most important aspects which has basically revolutionized our drug development workflow. The second is going to be characterization, which usually comes after the quality by design. Whenever you do a dosage form, we need to characterize it. So there are several physiochemical characterization methods which is available, which you all know very well, and it's outside the scope of this talk. So this is basically the scope of this talk today. So which is about the ACAT and the PBPK model. So ACAT and PBPK models are recent advancements in the domain of pharmacokinetics. And these are going to be, when applied, is going to be giving you very insightful, very important insights into the drug performance characteristics, and then can also be used as a very important tool for optimizing the drug development process. And then we can use this models as a feed forward or a feedback loop to the QBD so that we can further enhance our product development or target product profile of the product. And apart from that, the fourth step, which we always do today, is the validation of the PK model. So the in vivo characterization has not yet gone. So whenever we try to develop through QBD, characterize it, simulate the in vivo product characteristics. So this part, the simulation part, when you see last 10 years ago, was still done in vivo organisms. So we're using in vivo models to do the characteristics of our understanding the biopharmaceutical characteristics of our product. But today we can use powerful artificial intelligence based simulation tools, which can, to a very great extent, cut our effort in doing in vivo characterization, and then can be useful for a feedback loop to the QBD, and also can be used as a reference for validating the in vivo studies, which is essential for product filing. So therefore, this ACAT and PBPK models are going to be very important tools in basic product characterization, in vivo product characterization, which until very recently can be only done in organisms, vertebrate organisms like mouse, rat, or even beagle models. But with the advent of advanced artificial intelligence-based software such as Gastroplus, today we are able to do the product characterization in vivo uh, in a simulated manner, and we can cut lots of effort that is going to be required, specialized skill sets and effort that is going to be needed to characterize the product in a in vivo setup. So having said this, this of late has emerged a very important skill set in the drug development domain, and therefore it is very important for us to for us as researchers or aspiring researchers or people which is already in this business of developing newer drug products to know and understand the capabilities of ACAT and PBPK based models, so that we can adapt them and cut our workflow, and we can augment our workflow to a greater detail. So before we go into the actual part, I just wanted to tell some basic history, or we can say the manner in which we have progressed today. So there are three generations of pharmacokinetics. So first, the pharmacokinetics was, to put it in perspective, began in late 1970s from William Tyrrell, and then it has developed through generations. So the first generation models, which like non-compartmental analysis, are basically black box models based on hypothesis, empirical hypothesis, and they're not very accurate because the analytical methods which supported them was not also very accurate. And somewhere in the late 90s, early 2000s, the compartmental model and the ACAT models developed from the non-compartmentals. And then with the help of better mathematical models and biological and cheminformatics and metabolomic support, this has emerged into very actionable and rational mathematical models by which we can model and simulate and predict biological responses to drug molecules, either small molecules or large molecules. So therefore, this has come a very long way from a very simple mathematical calculation. We have come to a more sophisticated model and simulating approach somewhere in the late 2000s. And talking of now, in the last 10 years, the domain of PBPK, that is physiologically based pharmacokinetic modelings, has been all the rage, and they are increasingly being used. Not only used, they are also accepted as regulatory data by all major regulators in the world, namely US FDA, EMA, and the likes of it. So therefore, the ACAT and PBPK models are going to be very important, uh, toolkits or skill sets that we need to learn and imbibe and implement in our drug development workflow, because these are going to be the future. In the next 5 years, I see this domain to be having explosive growth, and therefore as teachers, we should know these tools, what exactly works, what are the exact implications of this, and we should know the nuts and bolts of this domain so that it's going to be easy for us in the self-development in the next years to come. So as we all understand, the PK model is nothing but a mathematical modeling technique for predicting the absorption, distribution, metabolism, excretion, in our language, the ADME of synthetic or natural chemical substances in humans or other animal species. So therefore, PK models are basically sophisticated mathematical models based on ordinary differential equations, ODEs, which is going to be implemented in several software tools, which is going to tell us what is going to be the likely absorption, distribution, metabolism, and excretion when we are going to administer a synthetic or chemical substance in a known quantity, in a known molar dose through a particular route in either humans or animal species. So this, when we contrast with the in vivo testing, we have come a long way, because this was traditionally done by using in vivo models, by using either mice or rat model, or even higher models like simian or dog models. But increasingly, people are resorting to the systems biology tools like PK models to do this, because there are several ethical issues which is being raised. And doing an in vivo experiment is not easy. It's not everybody's cup of tea, and it requires specialized skills and ethical permissions. So therefore, many people, when right now, what I mean by right now is the past 5 years and counting, are increasingly resorting to either ACAT or PBPK models to do their PK modeling, so that they have a very good idea about the in vivo performance characteristics of their product. So the important aspect of today's discussion, which we already covered in one of the earlier discussions on PK modern sim, is the advanced compartmental absorption and transit model. So this original idea was first proposed by Yu and Almedon in 1999. And this is basically an advancement over the compartmental model. Usually we know the traditional compartmental models are one compartment, 2 or 3 compartments. But this was a breakthrough which was proposed by Yu and Amidon because they understood the inherent restrictions of this compartmental models. So they proposed this advanced compartmental absorption and transit, which is known as ACAT for short. So this ACAT has set as a very important and as emerges a very important model in doing PK simulations and getting to know the in vivo performance characteristics of drug products and drug substances. So therefore, what is the advancement which is there in ACAT over the traditional compartment models? So this ACAT model basically is GI oriented. The ACAT model has depicted the GI set of compartments. So earlier, when you contrast this fact to the normal compartment models, the GI is not considered a major compartment. But here, GI, these are going to be having different segments of the digestive tract. Stomach, duodenum, jejunum, ileum, and colon are considered separate compartments. And it is going to be giving you a very mathematically consistent and statistically consistent, a very realistic ADME profile of a drug, so that it is easy for us to mathematically model and get credible and tangible, statistically tangible result out of this model. So when you see this, many people's and many research companies, drug development companies are increasingly resort to this ACAT model, and they are developing products based on the ACAT and PBBK based modeling and simulation. So one such product which is being used in industry is Gastroplus. So Gastroplus is a licensed trademark of Simulations Plus, which is a US-based company. So they are into this software development business, catering this particular segment in drug development for the past 30 years. So this software is one of the few softwares, apart from Winonline, which is being used or which is being accepted for regulatory filings. So therefore, it assumes all the more importance because many of the major pharmacos, top 10 or 10, 15 pharma companies in the world are starting to use Gastroplus, and they are giving the PBPK data for dossier filing, and it's accepted to a greater extent in the last 5 years. So therefore, getting to know this ACAT and PBPK models is increasingly becoming imperative for us because this is going to be the way forward in the drug development apart from the QBD paradigm. So therefore, I find this topic to be having a very big relevance in the years to come. So as we all know, the Gastroplus is a complete package. So what I mean by complete package is that Gastroplus has end-to-end capabilities, right from starting the predicting the ADME profile of NCE to doing sophisticated AI-based ACAT and physiologically based pharmacokinetic models based on not only humans, but all higher order primates and even rodent models. So therefore, this software is being increasingly used in the drug development and characterization workflow across all major companies in the world, especially all the top 15 companies in the world, whichever is in the blockbuster range, which is having a revenue of more than $1 billion or more, are increasingly resorting to this software for their normal workflow. So hence, this software in particular is one of the important tools to know and also practice for our normal drug development workflow. But it comes with a caveat, it is not a free software. So many people which are academics will ask me the question, which obviously begs the question, what about, is there any free models which is available? Yes, there are several free alternative software which is available, but with lesser capabilities, and it's not as polished as Gastroplus. I can say there's PKSim by Open System Pharmacology is a very excellent alternative. You can also try that. It's 100% free. It comes in the open source software product, which has similar capabilities to Gastroplus. So today, we'll be concerning ourselves only to the topic of discussion, which would be the PK modeling of targeted drug delivery and novel drug delivery systems. So as we all understand, Gastroplus has 3 different levels of capabilities: basic, advanced, and add-in capabilities. In the add-in capabilities, there are several things which can be done with add-in capabilities, like optimization, IVC, IVC, metabolism and transporter module, PK plus, PD plus, PBPK plus. We can do ADMET prediction. And apart from that, there is a separate add-in which is exclusively used for targeted drug delivery and NDDS products. So today we'll be having or doing a major discussions on this. So as I've told you, there are several ways when you do a quick literature survey. There are 4 or 5 major ways by which the tests are designed. So whenever you see this manner in which the targeted drug delivery or the novel drug delivery are going to be administered, they are going to be either done by ocular route, or basically targeting the lung, or applying them on the skin or delivering the drug directly to the oral cavity. And apart from that, it can be either done by intramuscular or it can also be given by subcutaneous. But whenever you do a literature survey, you will find that majority of the targeted drug delivery routes, the most popular are four routes, which is the ocular, pulmonary, transdermal, oral cavity absorption, and transit model. So this models are going to be, whenever you try to model the drug or eye, is called as the OCAT, ocular compartmental and transit model, OCAT. Or if you want to model the drug delivery to a pulmonary lung, it is going to be called the pulmonary compartment absorption and transit model. Or if you want to do a transdermal based approach, this is called as the TCAT. Or if you are going to directly give the drug to the oral cavity, this is going to be called the OCAT. So these are going to be the four compartment and absorption models, which are based on ACAT and also increasingly reliant on PBPK and artificial intelligence, so that it can efficiently model the drug absorption through a particular target site. And then we can do the pharmacokinetic modeling of how the bioavailability is going to be present from that target site. So this is going to be the scope of this talk today. So from here, we'll be going on to the hands-on part of the study. For this talk, I've chosen three case studies, which is going to be based on three routes of admin. First is going to be a PCAT, pulmonary compartment and transit model, which basically tries to model a drug delivery as a inhaled powder. And I'm going to use tobramycin as a model drug using Pulmosphere. I think many people can go and Google what is Pulmosphere is. Pulmosphere is going to be a drug delivery device which can nebulize and can suspend the particles to a very smaller. So this is very frequently used in countries like US, where it's a patented product. So what we're going to do in the first case study is that we'll be using tobramycin as an inhaled powder, where we're going to target the lung, and we're going to see what is going to be the percentage of drug absorbed pending a pulmonary administration of drug. So this is going to be the basic scenario. So we'll also understand that the powders are not only the manner in which the drug can be administered to the lungs. So there are several ways the drug can be administered through the nasal pulmonary system. So the drug can be inhaled in liquid or a solid state. So the drug can be basically given in a liquid or a solid state and can be delivered as a single dose over a period of time, or delivered as a single bolus dose into specifically the trachea, or nasal powder dosage form, which can be directly dosed into the nasal compartment in liquid or solid state as a single bolus dose. So therefore, the PCAT supports three kinds of drug delivery. One is that it can support inhaled, liquid or solid state, that is one. And it can support the single bolus dose delivered over a certain period of time. And then it can model a single bolus dose to the trachea. And apart from that, one of the most popular ways many people resort to in research, drug development research, is by formulating them as nasal powders. which can be directly dosed to the nasal compartment, and it can go through as a liquid or solid state, and it can be given a single bolus dose. So in this present case study, what we are going to do is that we'll be using tobramycin as a model drug, which is going to be used inhaled with the help of pulmosphere. Pulmosphere is a drug delivery device which basically used for nebulization of inhalable powders. So you can Google and find out what is pulmosphere. Pulmosphere is increasingly used in countries like Europe and US, where it's a licensed medical device where we can use it for nebulization and administration of drug through a nasal route. So I'll be just switching on the video feed so that I'll invoke the software and start showing demo the actual process. So in the screens right now, we are seeing the actual Gastroplus software. So this software is thanks to a friend of mine, which works in the industry, has given me this laptop for demo purposes today. I don't want a licensed copy of the software for information. So this is a fully loaded version of the software, which has all the additional modules so that it has become easy for me to demonstrate the capabilities of Gastroplus with some 3 TDDS examples today. So many of you are meeting the Gastroplus first time, so I'll just show how this software is organized. So this software is basically having 5 tabs. Okay. So first tab is called the compound tab. The second tab is called the gut physiology. The third is called the pharmacokinetics tab. Fourth is simulation, and fifth is graph. Okay. So this is just like having a wizard interface. Wizard interface means whatever inputs which is going to be given in this workspace is going to be carried forward to the next space and so forth. And the final simulations and graph, which is going to be coming out of our model, is going to be shown in this tab. Okay. So how are we going to start a typical workflow in Gastroplus is that we need to import a structured file. Okay. So there are several structured files which is available like 2D, inches or MOL or SDF files. So what we need to do is that we need to either go to certain sites where this structure files are available, or if you are working in your lab using a software like MOE or Schrodinger or even ChemDraw, you can also generate your own structure file to the extensions of MOL and SDF files. For example, if you are going to use, or if you are in the process of developing a new chemical entity, if you want to understand how it works, it also works fine in this scenario. So what you need to do is that you need to create your NCE in the form of a MOL or SD file so that you can import into the Gastroplus database. So what I have done is that I have taken the drug tobramycin. Tobramycin, as we all know, is an antibiotic, very good pulmonary antibiotic. So what we need to do in today's requirement is that we work as a powder if you are going to administer through a pulmonary route. So how are we going to do it? So as I've told you, I've already loaded the tobramycin structure file. So whenever the tobramycin, any structure file is imported into the Gastroplus, I'll just show how a structure file is imported. So you just go and import a structure. Okay. So whenever you import a structure, there is a thing called ADMET predictor, ADMET predictor. So which is a part of the software, which is going to predict all the physiochemical formulation properties and absorbed properties based upon the chemical structure of the drug. So whenever you import a inches file or SDF file or MOL file, the software itself will have the capability of predicting to a very high degree of accuracy all the physical chemical properties, formulation properties, and absorbed properties. And apart from that, we can also specify the pharmacokinetics and physiology of the drug. So this is how we basically do it. So whenever we try to import, we import it to a database file. And this database file, we're going to do some simulations based on this. So this is how the basic uh, workflow is going to happen in the Gastroplus. So coming back to our discussion, in today's aim is that we want to understand how tobaromycin is going to work if you're going to put them or administer them through the pulmonary route of admin. So what we're going to do is that I have just created a tobaromycin database here. So I'll just create a file. So here I have created a tobaromycin. And then I made a copy of the existing file here. So you can just see that's the same structure. So what I need to do is that I have to choose the dosage form. So as you have seen, there are several kinds of dosage forms, several routes of dosage form, which is supported in Gastroplus, like IV, bolus infusion. And apart from that, there are several ways of solid compressed dosage forms like immediate release, delayed release, controlled release. And apart from that, we have several ocular delivery models, which is given by the code OC. And then there are several pulmonary release, which is given by the code PL. So this is the matter of interest for us today. So in the pulmonary release, the drugs delivery system can be either a solution, a powder, or nasal solution, nasal powder. There are 7 or 8 variants by which we can model the release of the dosage form in a simulated environment. So in this case for us today, we are going to do it as a powder. So what we're going to do is that we are going to do select the option PL powder, because this is going to be a pulmonary release. So we are basically going to take a tobaromycin powder, put it in the pulmosphere, nebulize it, and then inhale. So after that inhalation, we wanted to know how much of the drug is available in the systemic circulation. So this is the basic scenario. So what we need to do is that first we have already imported the TOBRA structure file. So based on that, the ADME characteristics are already simulated into the database. So here we are going to take the pulmonary powder, and then we need to put the dose. So I'm going to give 80 mg as a dose. So there is no subsequent dose, and it's going to be a single bolus dose. So there is going to be a 0-hour dosing interval. And the most important part is that since we are going to administer this drug as a powder, it is important for us to mention what is the particle size of the powder is. So this particle size of the powder can be gotten from the physiochemical characterization whenever you do the drug development. So there are certain important things that we need to understand. We need to put the mean particle size in micrometers. So whenever you do a particle characterization, we get this data from any particle size measures like measuring techniques like Malvern or whatever. They basically give you the data in micrometers. So in this case, I found out from the actual paper, it has around 1.7 micrometers as the mean particle size, and the standard deviation was found to be 0.8. So I'll just put this value here as 0.8. And then coming to the number of bins. So number of bins is basically the sophistication of the particle size analysis. For example, the particle size technique works upon visual segmentation in most of the cases. So the number of bins is going to be the sophistication or number of visual segmentation bins available for the particle size. So in this case, there are several techniques depending upon the particle size analysis. That might be several bins ranging from 1 bin to 8 bins. You can ask the person which gives you the data. So in this case, I found out it's going to be having 3 bins. So I just enter this and then I put OK. So we have coded the database for tobramycin as a pulmonary release powder in a single bolus dose. So initial dose is going to be 80 mg. And then we have just put the particle size to be 1.7 micrometers, having a standard deviation of 0.8. And depending upon the particle size segmentation is 3. So then we are going to understand how we are going to do. So Gastroplus, as I've told you, is a very sophisticated software. We can do several kinds of physiologies. So what I mean by physiology is that we can do a human model, a beagle, or monkey, rhesus, or cynomonkey, rat, mouse, mini pig, cat, human ETT models, whole lot. Okay, and apart from that, it also has models where there is going to be hepatic or liver compromise or disease model, which also can be done. So based on that values which are going to get, we are going, the ACAT value is going to set. So I have told you the ACAT value, the GIT is segmentized into different compartments. So as per the ACAT model, there is a stomach, duodenum, jejunum 2, ileum 1, 2, 3, cecum and ascending colon. So depending upon what physiology you're going to mention, for example, if I'm going to take a beagle physiology, you can just see it will automatically adjust itself. So it is a very useful. So I have just chosen a human fasted physiology. So in the pharmacokinetics tab, so I'm going to say it is a compartment model. Since I don't have any values, FA, FDP or percentage absorbed, I'll just leave it to the default value. So, and then I go to the simulation. So we are going to do a single simulation, or we can also use a population simulation depending upon a small population, and we can mention the simulation length here. So I'll be going for around 12 hours in this case, or for example, we can also see what is going to be the simulated length for 16 hours. So having said that, what we are going to do is that we are going to go for, since it's going to be done in a nasal route, a pulmonary route. So what we're going to do is that we're going to open the PBPK model that is going to be associated with the pulmonary route. So here we are going to describe the PBPK condition in which we are going to operate this. So there are several ways of doing it. So the PBPK ACAT model that is associated with the pulmonary route supports 4 organisms, human, rat, mouse and dog. And then it also supports the gender. It can be a male or female. So as a default value, I'm choosing this as a human, a male gender. So it can be, age can be specified here. So here for this sake of simulation, I'm just giving a 30-year-old human male. And then this is the important part. So this basically PBPK, it is going to say how are we going to mathematically describe the disposition. Okay. So here, since we are going to give it as a powder, which will eventually deposit into the pulmonary matrix, there is going to be a deposition model. So there are going to be several deposition models. For example, there is a Finlay mouth model, IR ICRP 66, which is a very common model which is being used in this domain. You can read Hickey's if you have any doubts. So this would be a discussion by itself. So I'll just go with a user-defined model. So user-defined means if you're going to inhale the powder through your mouth, and it eventually will go and deposit inside the matrix of the lungs. So we need to specify how much of the drug needs to be deposited in which part. So I have, for example, I just wanted to understand the particle size characteristics of my, or the formulation characteristics of my drug. I have given a deposition model which is user defined. So I'm going to say how much of the drug needs to be deposited there, because in some cases there might be some enzyme systems or receptors in particular regions of your target organ where you will find the deposition to be having the best pharmacological result. So therefore, if you want the site specific deposition, we can mention the percentage wise site specific deposition. So I've just given extrathoracic to be 40, just a random value, 44%. Thoracic, I've given around 9 or 10%. Bronchular, at 9. It can be any random value. So I just give it OK. And apart from that, we can click any of the compartments and say or modify any part of your compartment so that you can have a great degree of sophistication in your simulation. So I just save it. And then we are going to run the simulation for 16 hours. So until now, we have taken tobramycin. We have set the route of admin through inhaled powder. We have given the input of the particle size, the standard deviation, and the bins. And apart from that, we have defined the human physiology. And then we have defined the PBPK with respect to the physiology of your lung. We have done a user-defined deposition. And here is what it is saying, how the predicted profile of the plasma concentration of your drug is going to be after 16 hours. So if you want to know more, we can see site-specific absorption. For example, I just wanted to see a pulmonary. So I can just see how much in extra thoracic, bronchular, pulmonary, or how much goes into the lymphatic uptake. So I'm getting an individual AUCs for everything. And then I get a simulation of how the predicted plasma concentration is going to be. We are going to give it in a powder form through inhalation route on deposition to certain regions of your pulmonary region, and we are getting a actual simulation. So this is about the first case study of the day. So second case study of the day, we'll be progressing. So I think your heads will be swimming in questions. We'll take as many questions as possible at the end of the session. Please keep your questions ready. So second case study is where we are going to go now. The second case study is going to be a transdermal reservoir patch. So I'll be just switching on the video feed to detail about the second case study. So the second case study is a TCAT model, transdermal compartmental and transdermal compartmental absorption and transit model, TCAT model. So TCAT model is primarily used for modeling the transdermal dosage form. Transdermal dosage form, there are going to be 6 different kinds of transdermal dosage form based on US FDA classification. So because many people, in my circle, I find many of my colleagues and friends are working on transdermal products with controlled release characteristics. So I felt that many people requested me to take this case study of bopidnolol, which is a very potent beta blocker. So what you're going to do is that we are going to take a controlled release formulation, which is going to be a reservoir dermal patch. So the reservoir dermal patch is going to be having a construct like this. So this is the drug reservoir, the drug release membrane, and then we are going to stick it onto the skin, and the drug is going to be absorbed or going to be transiting the dermal layer, and which will be going into the systemic circulation. So this can be modeled in several ways. Especially people prefer transdermal dosage forms because of this controlled release characteristics, because many of our compliance problems can be overcome to a great extent by formulating drugs as a TDDS. So coming back to our discussion, there are 6 different types of transdermal dosage form based on US FDA classification. So first is going to be clear and homogeneous liquid formulation. Second is going to be a suspension, which is either a solid dispersed in a liquid, or it can be an ointment where 20 to 50% is in dispersed solids, while a paste has more than 20 to 50% as a dispersed solid. So we have to understand there's a distinction between paste and ointment. Ointment has 20 to 50. It is going to be greater than 20 to 50. It's called a paste. Then we have a transdermal gel. So transdermal gel is a formulation in which we are going to have a colloidal dispersion with a gelling agent. Then we are going to be having an emulsion, which is a semi-solid formulation of more than 50% water and volatiles, or a semi-solid formulation which is less than 50% water volatiles. If less than 20 to 50% of the volatile is dispersed, either greater than 50% are solids or waxes, PEGs, HGs, or greater than 20% is water or volatiles. And lastly, reservoir patch. This is going to be a reservoir dermal patch, which is going to be having a controlled release profile into the skin. So for this case study, I have taken from a published study of reservoir dermal patch of bopidnolol. As I've told you, it's going to be a blocker. We are going to formulate this particular drug as a controlled release reservoir dermal patch so that it can allow the absorption of the drug in a sustained or a controlled release manner for a longer periods of time, so that we can just stick it on the skin and get the benefit of the drug for prolonged periods of time. So that is the scenario. So I'll be just switching the video feed to demo this case study. So what I'm doing is that I'm just importing this database file into our Gastroplus database. So you can just see this is bopidnolol. So whenever I import, automatically the system is going to assign you all the physiochemical factors, characterizations like molecular formula weight and log P, pH value, everything is done. So in this particular case, what we're going to do is that we are going to formulate this particular drug as a patch. So I've already made this file ready. So in the dosage form, what we need to do is that we need to put this as a reservoir patch. So reservoir patch, I've already told you the transdermal. There are several ways in which you can simulate transdermal drug release in gastro GP. So it is going to be either liquid solution, liquid suspension, ointment or paste, lotion or gel, cream or reservoir patch. So here we are going to do as a reservoir patch, and the initial dose, loading dose, is going to be around 14 milligrams, which I've already put it here. so that there is going to be negligible dose volume because we are going to stick it into the skin. So we are not going to use any fluid uptake with the drug. So we are going to just put a minimal value for this. And from the literature I have surveyed, I didn't have any particle size which is mentioned. So I'm not going to put, I'm just going to use default particle size value for this. And after this, what we're going to do is that we are going to go and model our transdermal view editor. So this is the PPBK model for a transdermal drug delivery. So here we are going to be having several options. The first option is that the region in which the drug is going to be placed. So in this case, the most common way is that people just put their transdermal patches under the arm. So there is human arm, back, abdomen, leg, because we have different sites, because depending upon the area we're going to put, the vascular residue is going to change. And therefore the absorption character profile is also going to be unique for each site of the physiology. So therefore, Gastroplus takes this into account. So there are several sites where we can do human arm, back, abdomen, leg, or human in vitro abdomen also is possible. Then if you're going to do mini peg ears, snout, neck, or also back is there. So for this purpose of study, we are going to take the human arm, and the skin pH needs to be mentioned. So usually we know the skin pH is slightly in the alkaline. So we'll be giving a default value of 5. Okay. So we'll go to this vehicle compartment. Vehicle compartment is nothing but uh, the manner in which we are going to load the drug. So this vehicle compartment here, we are going to define the characteristics of the formulation here. So as I have told you, the loading dose, initial dose is going to be 14 milligrams, and the dose volume, because we are not going to put any liquid fluid along with this loading dose, we are going to give a minimal value. And this is the most important part. For example, the total surface area, which is measured in square centimeters. So the surface area in which the drug is available for permeation through the dermal barrier. So here we are going to give it as a value in square centimeters. So this actually represents the total surface area in which the formulation will be applied. So we are going to give a value of 20, and then the application time usually in hours. So all this reservoir patches are designed for long haul, means that they'll be having enhanced times of application. So I have given somewhere in the region of 6 days. So I just okay it. Similarly, you can do different kinds of requirements for different compartments. So when you see a TCAT model, it has models for vehicle, the outermost layer stratum corneum, viable epidermis, dermis, and subcutaneous tissues. So each and every compartment can be defined. So here we can understand, for example, if there is going to be any condition, every human genetic variant is going to be having different skin types, moisture, and the partition coefficient of stratum corneum is slightly different across different races. The Negroes, the Negroid population, or the Caucasian, or whatever Mongoloid population, they have different skin makeups. So So the stratum corneum partition coefficient is very important. Diffusivity, stratum corneum diffusivity. So there are you can choose any one of them. So for this map, I can just choose the Robinson model, and then we just choose us. Then we need to go and define the control release aspect of this. So as we all know, this reservoir patch is going to be having a control release formulation. So to define the control release characteristics of this this module, control release profile module, is going to say how are we going to characterize this control release aspect. So here we are going to have a representative equation of how much dose should be released. So I have chosen 3 ways of doing this. There's going to be either single variable, double variable, or triple variable. So we need a long haul release. So by default, we will be knowing from the theory, double or triple variable is going to be a good aspect. And then we need to give what is the phase 1, phase 2, or phase 3, basically the fraction absorbed, the time scale, and the shape of the curve. So in the fraction absorbed, I'm going to say how much of the drug is going to be absorbed in phase 1 and phase 2. I just want a minimal amount of drug to be absorbed in the phase 1, so I put 0.1, and then the time scale hours, so which is going to be the definition of how much of the time that needs to be simulated. So here in the phase 1, I'm giving 5,000, and in the phase 2, just 1,000. And the B shape, I want a more sustained release, so I give the B shape larger number, and then I click OK, and then I'll go and I'll start simulating the percentage of drug absorbed for 216 hours. So if you could do the math, you'll know 216 hours constitutes 1 week time. So let's try to start simulating it. So the control release windows pops up, and then we already defined the control release parameter. So the simulation starts for 216 hours of the drug release from a controlled release reservoir. So the workflow we have done is that we have imported a structure file, defined the control release profile, and apart from that, in the PBPK model, we have defined where we have put the drug and the surface area of release, the initial dose, and apart from that, we have also defined the compartmental characteristic of stratum corneum and the diffusion profiles also. So if we are into the simulation, so we are almost there. So this 216 hours. So you can just see this is the transdermal patch plasma concentration. This is how it is. So if you want to know more the specifics of it, we can also see region-wise absorption. And then if you want to know more about, we can do a ACAT model, or if you are more interested in understanding the dermal aspects of it, we can just see how much of it has happened in particular compartments of our model. So we can just see the absorption profiles of compartment wise here. So this is about the second case study, which is a reservoir patch of a beta blocker of buprenorphine for a week's time. So this is about the second case study. In the second case study, we have just attempted a transdermal reservoir patch with a controlled release profile. So we will be progressing to the third case study. For this, I'll be going back to my PowerPoint slide. Please. So the third case study is going to be oral cavity absorption transit model. So this will be the third case study. This OCAT model is primarily used for understanding how the or modeling the pharmacokinetics of drugs pending oral cavity absorption. So the oral cavity, as all know, is the when you whatever physiological features which is present in the mouth. So if you are designing a drug system, drug delivery system, which is going to be primarily intended to deliver drug to this compartment, then we can use this OCAT model for our requirement. So the case study I have taken is a very interesting case study, which is a marketed product called Actic. So Actic is a lollipop. So what I mean by lollipop is this. So Actic is basically a lozenge-based drug delivery system. So as we all know, fentanyl citrate is a potent opioid analgesic, which is especially used for pain management in cancer. So this unique product uh, is a marketed product called Actic, which is nothing but fentanyl citrate lozenge, which is available as a lollipop. So what we have is that we have a plug of drug in which is attached to a plastic rod. So whenever people will have a pain stimuli, what they do is that they take the drug out of the container and then they keep the the lozenge plug between the teeth and their oral mucosa, so that the drug is going to be directly absorbed from there, and then it is going to, the pain relief is going to kick in. So this is one of the hugely successful products, especially which is used for pain management, especially for oncological intervention pain management. So what we are going to do in this case study is that we are going to simulate this scenario, the marketed product of Actic, comprising fentanyl citrate, uh, as our case study. So before we go into the case study, according to the US FDA classification, there are 7 different kinds of intraoral dosage form, of which 6 are going to be immediate release and 1 is going to be controlled release. So the immediate release are going to be either a sublingual solution or tablet, supralingual tablet, lingual spray, lingual solution, buccal patch dosage, which can be used in a controlled release format. So intraoral route of admin is going to be a very popular route of admin, and I've seen many people working in this domain. But the problem is, if you want to characterize this with the help of an in vivo model, it is a bit tough to do that. And it requires lots of pharmacological expertise to do this. So with the help of this simulation model, a OCAT model, let's see how to do it more efficiently with almost identical result. So I'll be just switching on the feed. So I've just loaded the fentanyl drug file here. So what are we going to do is that our aim is to create an intraoral dosage form. So this is going to be an intraoral tablet. So the loading dose would be around 700 milligram. So it comes in two strengths. One is 700 or 1400 mg. In some cases, it also comes at 2100 mg. So the volume is, since this product is going to be intended to keep between the cheeks and the teeth and then allow the drug to permeate from there, and the saliva which is going to be secreted in the process is going to be swallowed in. So therefore, since there is going to be some secretion of saliva involved, we are going to put a minimal value of saliva at the dose volume to be 20 ml. So we'll just put it here. And then we need to, since I didn't find any particle size in the literature, I have just taken this as a default particle size. And then I've taken the gut physiology to be fasted state. And then I go to the oral cavity PBPK model, and then I'm going to model this particular compartments of our IO, intra-oral drug delivery. So here we have a oral cavity. So there are going to be 3 or 4 physiologies. We can simulate the oral cavity of a human, beagle, monkey, or rabbit. And apart from that, we can also do it based on the gender. And then we can specify the age. And then depending upon that, we can, some products are intended to be kept in mouth. So we can keep it as a awake state or sleeping state, depending upon the lucid state of the people. And the residual saliva, which is present in the mouth after swallowing it. And then we are going to be having a experimental setting on how to do it, how the transit is going to happen. For example, if you're going to do normal swallowing, or hold and swallow, hold, rinse and swallow, or hold and spit it out, or hold, rinse and spit. So any kind of situation can be simulated. So in this case, Actic is designed as a dosage form where you need to keep it between the teeth and the oral mucosa. So I have put the transit model as hold and swallow. I have put it a holding because as per the product literature, I found that the holding time is 5 minutes. So I've just put the 5 minute here. So if you are this thing, you can also mention the drug deposition in the regions like buccal cavity, gingiva, palate, tongue bottom, or floor of the mouth. So since I didn't have any data about it, I have just left it as default. So we'll be using a ACAT PBPK compartment modeling. And then we want to see the simulation for 24 hours pending a 5 minute administration intramucosal, from intramucosal route from the mouth. So we have just imported, given the condition, and then we have defined the oral compartmental and transit characteristics. And then we are going to do our simulation for 24 hours. Sorry, I just forgot to mention the deposition characteristics, sorry. So I need to, since this is going to be a product where the deposition is going to be happening primarily in the mouth, in the buccal cavity, since you're going to hold it between the teeth and the cheeks, majority is going to happen in the mucosal buccal region only. So I'm going to give the buccal region as 100. And afterwards, I'm going to simulate it for 24 hours. So it's done. So we can just see the absorption resolution and the CP time of the concentration, plasma concentration from intramucosal route of fentanyl for 24 hours. So we can just understand the PK characteristics here. So this would be the third case study and the final demonstration case study of today's talk. And I request the moderators to open the floor for questions.
Am I clear, sir? Audible?
Yes, sir, you are.
Yeah. Thanks, Dr. Chandramouli, sir. It was another nice session and very informative. Our YouTubers are very full with the appreciations to you.
Thank you, sir.
And thanks all of you also for witnessing a wonderful session, which we have mentioned that it will be always innovative. Now we are going for the questionnaires.
Yes, sir.
Please raise your hands if you are having any questions so that we can ask the speaker. Raise your hands, please. There was a question basically from Dr. Mahendra Rana.
Yes, yes, sir. Please ask a question.
He has asked that please share the open source softwares that can be used in place of Gastroplus for teaching learning purposes. If it is anything there.
Yes, sir. I have already told in the previous, if they could have seen my previous talk, there are quite a few open source software which is available. So PKSim is one of the software which I recommend using. It's called PKSim. Please Google for Open System Pharmacology, OSP.
I think Dr. Rana, you must have got your answer. There is another question from Arpana Patil. In case study 1 for pulmonary CAT, what particle size is entered?
I have put 1.7 micrometers.
1.7 micrometers, Dr. Patil. And geometric diameter or aerodynamic? MMAD.
Geometric diameter, sir.
I can see that raising hands option. Is there in the raising hand option, please? YouTube, any question you want to ask, please ask.
Sir, can I unmute, sir?
Yeah, you please, if you can visible to you, please do that.
Yes, Dr. Shashikant Singh, please ask a question, sir. Sir, Shashikant Singh, sir, please ask a question. You're unmuted, sir. Shashikant Singh, sir, please ask a question.
Yeah, Shashikant Singh, please ask a question. No, he is not answering. So we are going to the next one, sir.
Ajish.
Ajish Reed.
Ajish, sir, please ask a question.
Dr. Saroj Kumar Patro. If you yeah.
Very nice presentation from the sir.
Thank you, sir. Please ask a question, sir.
My question is, it is absorption, distribution, metabolism, and excretion regarding the pharmacokinetic parameter.
Yes, sir.
the study of pharmacokinetic parameter, how will monitor the concentration of metabolites in every step of pharmacokinetic parameter?
Yes, sir. It's a very important question to ask. So many of the drugs are either pro-drugs or having active metabolites. So we, in case if the drug is having a known active metabolite, it's always better to model with the active metabolite, sir.
Yes. Okay, sir.
Thank
Sir,
you.
actually, this is the only in vitro study.
Yes, sir. This is an in silico study actually.
In silico. So how will monitor the different dose concentration during the toxicokinetic study? Because when we are going to perform any toxicokinetic study, first we have to do the pharmacokinetic study.
Exactly, sir.
So what are the parameters to observe the various concentration of drug in plasma level?
Sir, this is basically based on, as I've told you in the talk, it's based on compartmental and transit model.
How to monitor the concentration of the drug during the toxicokinetic study?
Yes, sir, we can
Is it possible to do any toxicokinetic study?
Yes, sir, it's possible to do this.
So how to monitor the concentration of the drug?
So the concentration of the drug can be gotten as an Excel file as output. So which I have not shown for the reasons of derivative. You can take the Excel file and you can have a minute by minute or second by second toxic time profile to plasma graphs. It's possible.
Okay, sir. Thank you.
Thank you, sir.
Thank you. So another one question, sir.
Yes,
This
sir,
software
please.
is a free version.
No, sir, it's not available in a free version. I've got it from
Only,
a friend. Yes, sir. I've
only
got it
the
from.
paid version.
Yes, sir. This is not even my computer. I borrowed it from a friend to demo for this program.
Okay, sir.
Okay.
Okay, sir. Thank you.
Thank you, sir.
Very nice presentation from your side, sir.
Thank you, sir. My pleasure.
Thanks, Dr. Patra. Please unmute yourself. Dr. Mahendra Rana, if you, the same question you have put in the YouTube, it's okay. Otherwise, you can raise your question, sir.
Yes, Rana, sir. Please ask a question. Mahendra Rana, sir, please ask a question. Partha Pratim, please ask a question, sir.
Okay, Dr. Sakti's iPad. Dr. Sakti. Dr. Sakti, you can unmute yourself and ask a question. Okay. Sayani Bhattacharya.
Yes, Sayani ma'am, please ask a question. Sayani ma'am,
good evening, sir. Yeah, good evening, sir.
Good evening, ma'am. How are you?
Fine, sir. I have two questions actually.
Please.
It was a nice virtual experience of drug absorption from specialized delivery through virtual platform. The question on inhalation, first question on inhalation, you have shown the site specific deposition through nebulization or maybe through DPI, which is only a function of particle size. But actually, some other factors like particle density, surface area, surface charge, even hygroscopicity also plays a role in specific deposition. Just want to know whether the software considers the rest of the factors for actual simulation.
It considers the localized SIP and the target receptors present that can be modeled. That is
because
one.
Not only the particle size, the particle size only less than 1 micron, like 0.5 micron will be reaching the alveoli. So rest form will be depositing in the lungs. So whether that particular, like if I have to show the polydispersity of the particle, or if I have to see, like whenever we are taking the nebulization, or the particles are coming in the wet surface of the mouth, the hygroscopicity changes the particle size. So that consideration.
So yeah, I told you how that administration protocol is going to be, right? So I've shown you, I have used a user specific protocol. So there are several 3 or 4 manners in which the drug can be administered. So you can play around and find the correct aspect of your delivery to your particular problem. It can be done.
Okay, sir. Thank you, sir. Another question I have.
Yes, sir. Yes, ma'am.
That is on transdermal.
Yes.
Sir, you have shown that the Weibull factors, the shape factor, just in that case study, the shape factor for the double or bi-Weibull function study, it was generated from the in vitro kinetic studies, or the software will be generating, sir?
So you can basically, you want to have a particular release profile, right?
Yeah, if it is Weibull only, okay, let it be Weibull, or then because it can take by Weibull, or even it can go up to 3.
Yeah, it
So
can go to
I
the
just
function
want,
of 3, single, double, or triple Weibull can be.
yeah, single, double, or triple. So generally we can find out Weibull function in normal Excel sheet through the in vitro dissolution profile.
Correct.
So I just want to know, in the software when you have imported that Weibull function, was it from in vitro study, or the software generates, or
No,
how
it generates based upon the permeation characteristics. It gives the best fit.
Oh, the software only gives the best fit for it.
Yeah, exactly. Yeah.
Okay, sir. Thank you, sir. Nice session, and very interesting session also it was. Thank you, sir.
Thank you, Sainidhi. Thank you.
Chandramani sir.
Yes, sir.
I'd like to request you to stop the share screening, share sharing, share the screen, so that all you can be visible there. Thank you.
Yes, sir.
The next one question has come, sir, from YouTube. That is from Kamini Sharma. Is there any CAT for brain also, sir?
Yes, it's possible. If you want to do BBB, it's possible. You can.
Another question from Satya Bharadwaj. Sir, is it possible to calculate the protein binding of given drugs?
Yes, it's possible.
Now another question from Rajendra Vera, sir, whether it take every considerations or factors in determining the PK data?
Can you repeat the question, please?
Whether it take every considerations, factors in determining the PK data?
Yeah, it calculates all the PK parameters. That is.
So
if you were asking about the PK parameters, right?
Yeah.
Yeah, it can do. So by default, it reduces 22 PK parameters.
Okay. So you must be getting an answer. Gita Agarwal, please ask your question.
Ma'am,
you unmute yourself, ma'am. Ma'am, unmute yourself. Yes.
Yeah, I have done it. Thank you, sir, for very nice presentation. I have one question. As we are working on one of the formulation, that is solid lipid nanoparticles. So my question is that whenever we want to find out pharmacokinetic parameters of this formulation, are there other factors also, or some special points that should be considered along with the particle size for this particular formulation, whether it is for transdermal drug delivery or nasal drug delivery? So,
in the very first slide, I showed you about the paradigm of, so whichever factors that you have taken in the quality target product profile, you find which is going to be having a very important significance in the performance characteristics, those should be given leverage.
Right, sir. Sir, you have shown that that particle size, of course, we have to write down everything. But because whenever these kind of formulation they are worked upon, what happens? There is cellular uptake and other factors. They also matter in the effect of that drug. So in that case, should we consider some other parameters also for the accurate results?
Yes, of course. So not every drug is created equal. Only the researcher would know what kind of drug and problem he is working with. So I have taken 3 examples which I found in the papers which I found in the open domain, which I have demoed here. So it is up to you to understand, and because every formulation problem is unique, you have to do your own part. That would be the correct way to do it.
Yeah. Yeah. Thank you, sir. Thank you.
Thank you.
Very nice.
Mr. Radhesyam Samant, you are having a question. Please unmute yourself. Radhesyam Samant, if you are hearing me, please unmute yourself. Okay, Sarath West, please ask your question. Sarath West, if you are hearing me, please unmute yourself and ask your question.
Okay, sorry. Yes, sir. Yeah, good evening, sir. Sir,
evening,
as
sir.
the number of carriers are limited and it may vary, the number may vary. So can we predict the accurate transfer of drug by this software if drug is transferred by the facilitated or carrier mediated transfer?
Of course, it can be.
And sir, if as you know, this drug may be drug PK, depending on the PK value and meal fasting and non-fasting condition, there may be different degree of ionization. So can we calculate the transfer of drug on the basis of?
Yeah, sure. If it's going to be having more than 1 point of ionization, it takes into picture. It takes into consideration.
Thank you, sir.
Thank you.
There is another question from Mohammad Moyuddin Sarif. So please ask your question, sir.
Yes, sir. Please ask a question.
Mohammad Moyuddin Sarif, sir. Are you listening? Okay. Dr. Amit Kumar Sharma, please put your question forward. Dr. Amit Kumar Sharma.
Hello.
Yes, you are audible.
First of all, I would like to say thanks for this presentation. Right, we have a very good presentation which you have given to us. It is very informative sessions. And secondly, the things which I would like to ask you is that how much precise this software it is in terms of calculating the data.
Yes, sir. I just read a paper yesterday only. The TCAT model is almost within the predicted versus actual is within 2%, sir.
Okay. Thank you, sir.
Thank you.
Dr. Prabhakar RV. Dr. Prabhakar RV, please put your question. Dr. Prabhakar RV.
Yeah, please unmute, sir.
Prabhakar sir, please ask a question. Prabhakar sir?
No, he is, I think, not audible. So there is an interesting question from Dr. Ajay Kumar Tiwari.
Yes, sir, please.
From YouTube. Is there any online course in PK modeling?
Yes, sir. You can try ADX. You can try edX platform. There are, I found 2 or 3 biopharmaceutics and things. And apart from that, there are several courses in Udemy also.
Okay. There is another question from Muhammad Havan Akhtar. Please unmute yourself and ask your question.
Actually, sir, I would like to ask one question from my side.
Yes,
When we consider drug simulation using PK modeling, using some software, Okay, as what happened in case of optimization process of any formulation, like we are putting some independent variables before proceeding into the optimization procedures that could significantly influence the process parameters or the entire fate of the formulation. Can we similarly put some important factors that could predict the fate of the drug absorption ADME profile by considering PK modeling?
Yes, sir. Specialized kind of population for liver impairment, liver injury, or if you want to do it in pediatric population or gestational population, which is not otherwise possible, we can do it with this help of this tool.
Hello.
Yes, sir.
Sir, actually, I want to know, there are some factors that has to be critically considered before designing PK modeling using some software.
Exactly. So I really don't understand the question. Can you repeat any factor?
No, please repeat, sir.
Your voice was a bit breaking. I didn't understand it completely.
Okay, sir. Actually, I want to know, optimization of any novel formulation. When we consider optimization of any novel formulation using some software-based modeling.
So optimization means you mean the development process, right? The QBD process.
Yeah, development of a dosage form of a novel formulation. Similarly, we are going to simulate our drug ADME profile using PK modeling, modeling techniques using some software. So, sir, I want to ask in this, what are the main important points or what are the main important things that is need to be critically considered before opting that software, especially in PK modeling?
Sir, as I have told you, it will depend upon two factors: the drug you are working with.
Okay.
Second, you need to understand the complete physiochemical properties of the drug.
Okay, sir.
Then it would be formulation dependent.
Okay, sir.
So formulation dependent means then there would be a lot of formulation dependent factors that is coming into picture, like permeation.
Okay.
Particle size.
Okay, sir.
The physiology, fed, fasted, set physiology.
Okay,
so
sir.
There are, I can think around 20 or 30 different kinds of factors that will come into play.
Okay.
So,
thank you
so as a researcher, you know your product better, you know your research better. So when you are coming to a situation like this, you are the best judge.
Okay, sir. Okay, sir. Thank you, sir. Thank you so much, sir.
Thank you.
So, Sheikh Munir, please unmute yourself and ask a question. By the time Sheikh Munir will come back, so there is another question coming from Satyavrat Bharadwaj. If a patient has hepatic disease, can we calculate the elimination half-life of given drug? Sir, am I audible to you?
Of course, sir. Because all this condition, disease state condition.
Important advantages of having this kind of lots of simulation, which is otherwise is not possible, because many of the diseased population data is not available to us. So in situations like this, we can use this kinds of tools to understand the disposition, especially in a disease condition like renal or hepatic impairment. We can.
So there is another question from Gunjan. I have unmuted you. Please put your questions. Gunjan, super. Okay.
Yes, sir. Good evening.
Good evening. Please put your question, ma'am.
Hi, good evening. Please ask a question.
Sir, actually, I want to ask, what is the scenario for the herbal formulation? Will it be the same?
A very important
Like we
question.
have isolated any compound herbal
Yeah,
phytoconstituent and then we want to go for the PK modeling.
Yeah, I'll tell my personal experience from this, from my this thing. This software works very correctly for small molecules, synthetic molecules. So if you're coming to natural products, if your product is correctly characterized, you have a correct structure, you can very well do it. So if the compound is correctly isolated and structurally elucidated,
Yes,
go with
sir.
this.
Okay.
Am I audible?
Yes, sir. Yes, sir.
Hello.
Characterization should be done in a proper manner. Then we can go.
Correct. If you're sure about your chemical structure, you can go rock and roll with this.
Okay. Okay. Thank
Thank you, ma'am. There is another question from Dr. Santanu Malik, and we'll be taking over to that last question, and then we'll go to the YouTube. So before asking Dr. Santanu Malik to come down, may I please put a request to all the participants, please mention your name when you are coming to the Zoom. Otherwise, the few numbers, just 10, 15 digits, we can ask for the question. So please, my sincere request, we can take your question, but we cannot ask you in the name of 9FE8D like that. So please put your name when joining the group, and ask the next question to the Dr. Santanu Malik.
Yeah, very good afternoon, sir. Hello.
Good afternoon,
Dr. Yeah, please, you are audible. Yeah, you are audible. You carry on.
Very much.
Yes.
Thank you very much, sir, and very good afternoon to all of you, and thank you, the expert, sir. My question is, can you suggest a few best references or resources or databases to check for drug metabolism pathway?
Yes, please check CHEBI, CHEBI.
Okay, that is a references, resources, or databases.
That is a database.
HEBI, okay.
And then you can go Red Bank CA.
HEBI, okay.
Red Bank CA.
And few extent, is there an Clarivate like that, something that can help Clarivate?
No, many of this is available in open source database. You can just search. But CHEBI is one of the good places to search. In fact, all the drug molecules have taken it from there.
Yes.
The drug structure files. Yeah.
Is it, sir, free? Is it free, sir? Free of cost, or it's there's charges are there?
Free. It's open source.
Okay. Yeah. Thank you very much, sir. Thank you.
Thank you.
Thank you. So there is a beautiful question which has come, and I think you are the right person to answer that.
Sir.
Yeah, am I audible to you, Chandramani sir? Yeah.
Yes, sir. Please.
The question is from Satyabrat Bharadwaj once again. If we can predict all the things from PK modeling, then what is the necessity for clinical trials? I know you are laughing because this was my question in the last webinar.
Yeah. So it's a very loaded question. So many people always doubt the integrity of this validity of this data. Rightly so. So many people complex phenomena to mathematically simulate and predict. But that's not the case. In the last 5 years, majority of the drug regulators like US FDA, EMA, or even TGA have started accepting this simulation data sets as a part of their filings. So it means that it is going to be a correct data set, and many of the published works, if you could read, the actual predicted values from the software and the actual PK, which is obtained from an in vivo model, is having a deviation of plus or minus 2.5 to 5%, which means that it's almost a very clear, prediction about human physiology is concerned. So then why clinical trials are required? Because the human genome is too complex to be, as of today, we don't have, even though we have a complete human genome decoded, the inherent factors, variabilities across the genomic patterns in all the human variants, genomic variants are too complex to be, done in a virtual screen. So that's the reason why people still are going for clinical trials, and still the in vivo data is considered gold standard. So where do these simulator tools are going to come into picture? The simulator tools are going to give you a very good, a best compromise approach. So as you see, the difference between a simulated result versus a in vivo result is not very great. It's plus or minus within 5% in most of the cases. So it can be used as a ballpark tool. We can understand where exactly are we proceeding, and it is going to be a good course correction tool. So therefore, it is going to be a very helpful tool for drug researchers before even going into in vivo results, making costly mistakes, and then restarting. We can know whether we are there in the picture or not very early.
Thank you, sir. It was the answer. I think sir has got it. I'd like to. It's a pleasure that Dr. Mohammad Salaluddin is on the line. We'd definitely like to hear from him two words. Yes, sir. Dr. Mohammad Salaluddin, sir. Okay, maybe not getting to him. So we are coming to the end of the session, and I'd like to ask Dr. P.N. Murthy, sir. Dr. P.N. Murthy, sir, he's you can say that sir is the father of pharmacy of Odisha, and all these associations, all these professional activities throughout his life, he has made it available for us till now, and he has sacrificed a lot of hours of time, and still now he is very active, and we are very thankful, sir, to find you. So we just want to hear you, and after that, I think your questions are one more question will be taking, sir. Welcome, sir, to the EFI Apti Odisha. It's a pleasure to find you, sir. Sir, your sound is not coming, sir. Sir, please unmute yourself. Professor Murthy, please unmute yourself, sir. Yeah, now it is audible. Please come. Hello. Yeah, it's audible. Yes, it is audible. Yes, Professor Murthy, it's audible.
Thank you. Thank you very much, Dr. Transdermal, sir, for your nice presentation.
Thank you.
You are really benefited a lot. And you have been contributing a lot through number of seminars throughout this lockdown period. And I also appreciate the APTI central branch, central team, and the APTI Odisha team for their untiring efforts in educating and updating the teachers of the pharmacy of various states. Especially, I would like to appreciate and congratulate and thank Dr. Mihir Kumar Kar and his team for initiating this kind of very useful activity in the state and helping all the teachers of the state. So I request all the teachers to make use of this opportunity and keep it up, Dr. Mihir and other team members. Thank you very much.
Thank you, sir.
Thank you, sir. Thanks for your nice words, sir. There is a last question from Dr. Suryakant Swami, sir. Dr. Suryakant Swami, please unmute yourself and put your question forward. Dr. Suryakant Swami.
Hello.
Yeah, please put your question forward, sir.
Yes, hello, sir. Good evening, sir.
Good evening.
Good evening, sir.
Good evening.
It is really nice presentations and nice informations regarding the software used for PK modeling. But one question, sir.
Please.
Before selecting any PK modelings for any drug delivery system, how you are assessing the critical process parameters, and how you are estimating the percentage of interstitial perfusion studies by using this PK modeling?
Okay. A very pertinent question. Thank you for asking.
Welcome, sir.
So it basically depends on what kind of problem that we are working with. So in the first case, in the case of pulmonary case study, which PCAT case study which I did,
yes, sir.
Particle size is a very important aspect. That is
okay.
one, and also the deposition parameter is another important aspect.
Okay.
So you need to know what kind of formulation
critical process parameters.
Exactly.
Yes.
So you need to basically translate what your critical process parameters will do in vivo. Okay. So you should have that knowledge before even you attempt a PK model.
Okay, sir. Okay. Thank you,
so
sir. Thank you so much.
You need to translate your physiochemical aspect knowledge into a biopharmaceutical
aspects. Okay.
Yeah. Vision. Only then you'll be having a correct answer out of this.
Okay, sir. Thank you so much. Nice presentation. Thank you, Dr. Meir, sir, for your nice initiatives. Thank you.
You're always welcome, sir. Thanks a lot for your question. It was nice presentation today and a fantastic session. I think you all have experienced it. There is tomorrow at sharp 4 o'clock. We are going to start orientation for pharmacy teachers in educational methodology, another beautiful session. So please stay tuned for tomorrow. And till that day, I think it is the time to conclude. But before the conclude, I would like to thank Chandramouli sir once again for your cooperation to us and dedication in preparing slides and softwares, taking a lot of hard work. And you have really entertained in our knowledge and with the intellectuality also. Thank you, sir. Thanks a lot. So I think.
Thank you, Miguel, sir, for having me. It is a really important effort that you have done. EFA is a very important visionary effort that you have put from Apti Odisha team. We are really thankful for you to host this program. And especially after this, even a very important catastrophe which happened in a state, you have come back with lots of energy to have this thing organized. Thank you, Miguel, sir. Thank you for having me. Thank you, Apti Odisha and Central team.
Thank you, sir.
Thank you.
So, dear participants, see you tomorrow at 4 o'clock with another innovation, with another innovative talks. Stay tuned. You will be getting all your link in your mail, in your SMS, and if not found, then we are having a YouTube channel. Please subscribe that. And our Facebook page is the Information Center. So any problem, you visit our Facebook page, ask for a chat, you will be answered immediately. Thank you, everybody. Thanks a lot. Next call of the day.
Thank you.
Over to host.